Hello, my name is Andrew, and today I'll be presenting on salt energetics as on the behalf of CC iGEM 2022. And yeah, so cell energetics, sort of how energy is handled in your body. How does your body utilize energy? How does your body sort of change the form of energy, and so on and so forth. So some prior knowledge that we want you guys to know. So just some biochemistry terms, sort of how the digestive system generally works, cellular respiration, photosynthesis. You don't really need to know any specifics right now. Just know what it does, what each one does. So just for a refresher, cellular respiration is essentially just utilizing some simple sugars and carbohydrates that you consume and converting into ATP, which essentially stores energy that can be easily disposable and used. And photosynthesis is actually the opposite, because instead it uses these like quickly disposable energy in order to create more complex structures like carbohydrates that they can use for storage. And both are very unique in their own ways, but you could sort of understand that they work backwards in ways, but not in others, which we'll talk about more in depth. So the first thing you need to know is about the sort of amino acids and sorry, not the amino acids, I mean the macromolecules in your body. There's four types, only three are displayed because someone changed my slides, but it is okay. So there's the carbohydrates, proteins, lipids, and nucleic acids. And each one has their own structure and each one has their own thing. I think another video will explain how each of these are structured, but for now, we're just gonna be talking about these very broadly. So what you need to know from this is, in order to understand the rest of this presentation, is sort of how carbohydrates work. They're obviously for energy storage. I mean, it's not energy storage, sorry, energy use. And for proteins, they're involved in a variety of ways, including strength, but also like there's a type of proteins called enzymes, which also help catalyze these processes related to growth, repair, etc. So that's why people say it's important to eat your proteins and etc. And that's probably the reason why. And so just sort of knowing about acids and bases, I don't think it'd be too relevant. Again, these slides have been altered in ways which I wouldn't prefer it, but essentially what you need to know about acids is that acids especially is that usually you can identify acid by seeing sort of a lot of a high H plus concentration. The higher obviously mean the more acidic, and the same way the more hydroxide or OH sort of molecules or ions in a substance, the more basic it becomes. And of course, hydroxide ions are negative. Okay. And then knowing what ADP and ATP are. So they're consisting of a pentose sugar, which is a five sugar, which is like a pentagon, hence the name. In this case, adenine is right here, which is the form of nitrogenous base, which you also found in DNA. If you're not sure what that is, you can go check out that video, which should be on the channel. But the important part of this molecule is actually these three or two phosphate groups, and phosphate groups are their own functional group, and they're sort of treated as like energy units, but it's not actually the phosphate groups, it's the bonds in between. So each phosphate group has like a bond to each other, which contains energy or potential energy or the ability to do work. But they can't because of their position. So basically think about this with analogy. You have a warehouse full of boxes with a lot of machinery that you can use at your factory. However, they're in boxes, so you can't do anything about it until you sort of take them out and sort of use them directly. And this is the same way. So you have a lot of stored energy in these bonds of phosphates, but they're not very useful until when you decide to like break one of these bonds and release the energy. And that's what the body does. It regularly goes in between. So it uses ATP as a sort of source of quick usable energy. And the ADP is, when we lost the phosphate from using the energy, and then you can repeat the process of making more ATP for the future, and etc. And there's like complex processes that goes into balancing these two. There's some regulating systems in your body, but for now, that's all you need to know about ATP and ADP. So just a brief overview about the uh, digestive system. So first is your mouth. And your mouth contains a sort of enzyme called amylase. So enzymes, as said, was a special type of protein that catalyzes certain chemical reactions, which includes the breakdown of substances. So in this case, your food goes in your mouth. You have teeth, you have a jaw, and some sort of mouth muscles. And what all of these do is they work together to try to break down large pieces of food. So that's already a first step in sort of microscope, like making it more micro, like so converting from like food to nutrients. And what these things do is they break down the food, and then sort of your jaw movements and combined with the amylase from which helps to break down food also from your saliva help to arrange these things in clumps called boluses. uh Ideally it's usually like a ball, but not always, depending on how thoroughly you chew your food and various other factors. But basically that's how the first step of how nutrition is obtained. And there's some specialized sort of adaptations with the mouth and sort of like your digestive system from the start, and that includes the arrangement of food into clumps or balls of food known as boluses. But also there are like in your esophagus, which is your food pipe, there are actually sphincters, which are just sort of segmented things. Think about it, it's like a centipede, but it has legs every like, I don't know, like 10 centimeters, for example. Same way with this, it just sort of helps the movement of food down. It's basically like flaps. And there's also your epiglottis, which is essentially like that hangy thing that you see in your throat that little kids like poking to try to see what it is. But essentially what it does, it just blocks the windpipe whenever you're eating. So when you're chewing, it knows and it sort of auto like goes back and blocks the windpipe. And this is to prevent mass choking, as there's sort of from your mouth, there's like two entry points, one into the esophagus and one into the windpipe. And obviously you wouldn't want to suffocate on food. So that's a sort of very interesting adaptation. Um, so in your stomach, there is a variety of layers, each with their own sort of function. I won't go into too much depth with these, but essentially it's very protected. There's many layers in your stomach, and what this does is to prevent sort of like stomach damage, because your stomach is very acidic, and the reason why is because sort of this acidity allows for sort of proteins in your body, in your food, to degrade. And uh like proteins are required and have to be denatured by breaking up the bonds, and this is done by putting in a very acidic climate that's like not to the ideal sort of pH of the proteins, and thus these bonds in the protein structure is broken down, and it becomes more simple proteins, sort of peptides that can be absorbed separately. And so now, as you can see right here, so in your mouth, you sort of start like the amylase starts breaking down the carbohydrates. So in the stomach, it breaks down proteins. So let's not get mistaken, it's not carbohydrates or anything. And then there's various types of specialized cells that I wish I could talk about, but obviously we need to continue with the video. But essentially these do a variety of things in helping to maintain chemical composition in your stomach, pH, to replace cells on the lining of your stomach, etc. So sort of the pancreas and the liver sort of work together in ways to ensure that pH is the appropriate. So bile production is found in the liver. There's a tube, there's a duct, I guess, connecting it to the small intestines. I mean, yeah, small intestines. And it secretes a substance called bile, which essentially just, it's like a very basic substance, sort of neutralizes the acid in the stomach. Obviously, after a while in acid, the proteins are denatured, and by if they don't neutralized acid, it's going to continue to flow down into your small intestines and sort of result in very bad outcomes that could be painful. And the liver also sort of like processes through sort of nutrients in your blood as well. Also, although the kidneys also do this, and pancreas is essentially another sort of organ that sort of helps to neutralize and to secrete the correct hormones and chemicals to continue digestion in the small intestines, and also regulates blood sugar as well. And so the small and large intestines is essentially where things like lipids, more carbohydrates are absorbed. Small intestines is very long, but it's like bunched up into your stomach. It's just a network of tubes that absorbs through diffusion, which we will talk about later after it's been hydrolyzed and absorbed back into the body. And sort of the large intestine sort of just dehydrates the food, resulting in the feces that is quite normally dry as compared to what you'd find in the stomach. And it also contains sort of sphincters, which help to make these shapes of feces that are stored in the rectum, which is where your feces are held before they're dispensed by the anus. And I forgot to mention that there's actually another adaptation in this peristalsis, which is essentially just rhythmic contractions of these sphincters in order to make the food move. Sorry about that. And yeah, so now let's talk about cellular respiration, sort of the main sort of section of our presentation. So there are many ways in which different macromolecules are sort of consumed. And so and sort of digested and sort of processed in order to make nutrients more accessible or usable. And there's many different processes and key steps in each of them: beta oxidation and oxidative degeneration. There's also alpha, gamma oxidation. You don't really need to worry about those. They were only going to be talking about how carbohydrates are utilized. And here are some important things that or keywords that you might need to know. And this is the general process of it. So the first thing is glycolysis, and essentially this step is the only step in cellular respiration. There are the conventional 3 stages of cellular respiration that doesn't really require the presence of oxygen. And that's because it doesn't use oxygen as a reactant to form pyruvate from glucose, and thus it can occur on its own without oxygen. So essentially what this does is each of these steps has a specific enzyme, which again helps catalyze this process of movement in between these different intermediate forms in order to reach the final outcome. Now, as you can see here, hexokinase, which is the first enzyme or in the first step, which helps catalyze it from glucose to glucose 6-phosphate, actually consumes ATP. And so on the top photo, you can observe that ATP is actually being used, and that's because this is the energy investment step where ATP is actually used. Obviously, cellular respiration, the end goal of cellular respiration is an attempt to gain ATP, to create ATP from carbohydrate structures, but this is just investment step. But as you can see here, a very key reason why ATP is needed here is because, as you can see, a new bond is being formed with the phosphate. And when this happens, you sort of need to input energy in order to form new bonds, right? Consider each bond like a sort of currency for the amount of energy held. So this is why in this case, ATP is needed in order to form this. And again, phosphofructokinase, which is a very important enzyme, actually the most important step, I think, in glycolysis. Again, there's another phosphate group that's added right here. Now, you might be wondering why. Hi, phosphoglucoisomerase. Why does it not require ATP? Well, because this is simply the hydrogen moving from this second carbon in the chain to the first one. And so this happens, there's actually like, it splits into 2 molecules, which then each sort of undergo their separate to create pyruvate. And essentially it results in pyruvate. So this is why glucose, which is a 6-carbon sugar, as you can see by the hexagonal ring, it becomes a 3-carbon, as you can see by this backbone. So there's 2 pyruvates made from 1 glucose. And this is actually the energy, I forgot what it's called, but essentially the stage where you get back the energy. Oh yeah, energy return stage, where you actually get back the ATP from your prior investment. This is in each sort of glucose invested, this happens twice, because as you can see here, split into 2. So there's 2 ATPs synthesized here, 2 ATPs synthesized here. So you end up getting 4. And this is like minus the 2 that were invested, you end up getting 2 anyways. So we'll be investigating this more into what phosphofructokinase is and sort of ATP accounting, but this is sort of the general process. You get pyruvate, which then undergoes pyruvate oxidation. This is a very short step, and essentially what happens is pyruvate, and this actually occurs, like glycolysis occurs in the cytoplasm, which is made of the cytosol, a special sort of substance, and sort of essentially through a transport protein, which are embedded on the sort of membrane of the mitochondria, it is able to enter the matrix of the mitochondria, or and essentially the interior. And now mitochondria are also, they also have a they have a bilayer, and that's to do with sort of the endosymbiosis theory that might be discussed in a later video, where essentially prokaryotes were consumed by other larger prokaryotes to create like these multicellular eukaryotes with like membrane organelles like the mitochondria. And so in the first step, carbon dioxide, where CO2 is removed, and a energy carrier, electron carrier, NAD plus, is actually protonated, or it gains electron, and this allows it to sort of energize and sort of help produce, or like help a later step function. And with the coenzyme A, it ends up being acetyl-CoA. And the acetyl comes from acetate, which is essentially when you remove the CO2, you pyruvate loses a carbon, and pyruvate's 3 carbons sugar. So this acetate becomes a 2-carbon sugar, and with the CoA, it becomes acetyl-CoA. And so then in the Krebs cycle, acetyl-CoA goes in and sort of binds to already 4-carbon oxaloacetate, and it forms a 6-carbon citrate with the CoA. And so it sort of cycles around and recycles itself. And remember, all 3 steps of this are in preparation for the sort of the main sort of big electron transfer chain. And sort of the carbons added here, as you can see, it becomes 4. So when you put in the acetyl-CoA, it becomes a 6. So how does it become a 4, and why does it happen? Well, again, I said that bonds hold energy. And so by removing a citrate here to become carbon dioxide, you're essentially sort of releasing some energy that helps in a form of electron that helps you sort of give an electron to this electron transport carrier, which can later fuel the electron transport chain, which generates the most ATP out of all of these steps in cellular respiration. And so a question that might be asked here is, why is there a CO2? And this CO2, the carbon comes from the 6-carbon chain, it becomes 5, but the oxygen comes from sort of respiring, so like breathing. Like when you breathe, you give your body a supply of O2, which helps to fuel this process at a like larger, more synchronized like scale with all of your cells in your body, which is like trillions, right? And so it becomes a 5-carbon, and then it becomes a 4-carbon after it when the same thing is repeated. Here again, the 4-carbon helps to sort of, these steps are also like energy given steps. GTP is actually ATP in terms of how it carries energy. The only difference is GTP has GTP has guanine instead of adenine as its nitrogenous base, and it sort of phosphorylates GDP, and it also gives another electron carrier, FADH2, which is just the form of the electron carrier with electron and a different type of electron carrier, and I'll explain it in depth later. But I return to this. So you're sort of recycling a 4-carbon that's already there, and sort of the electron transfer chain, or the main step in cellular respiration. It's essentially the most ATP generating stage. I believe it generates 32 ATPs as opposed to 2 in the Krebs cycle and in glycolysis. So in the electron transfer chain, what happens is a series of redox reactions occur, in which energy falls, but like this powers the rest of the process, which I'll explain later. So this sort of graph explains each of the structures in the electron transfer chain, which is embedded in the inner membrane. Again, I said that there's a double membrane of the mitochondria. So this is the inner one, closer to the inside. And there's these protein complexes that help to run these redox reactions in which energy is transferred and released in order to power sort of some of the functions here, which essentially create the ATP. And so NADH is the electrons carried by NADH, as mentioned from the Krebs cycle and glycolysis. It's given in complex 1 to this sort of complex, and it sort of goes down. So ubiquinone is right here, and this is important because this sort of regulates whether electrons are allowed through FADH2 on complex 2, like its electrons carried from the Krebs cycle. And so the difference between FADH2 and NADH is just essentially just a differently structured electron carrier. Interestingly enough, FADH2 is FADH2 is carries less energy with its electrons, so that's one thing to know. But essentially, why you need oxygen is because it can't be powered without FADH2, and since FADH2 is in the Krebs cycle, it requires oxygen in order for it to be given electrons. And because of this, in a state without electrons, glycolysis produces or gives electrons to NADH that doesn't do anything. And this is when fermentation comes in, as I'll explain in a second. So it moves down and through a variety of complexes. And as you can see, protons are moving through it. And this is through sort of active transport or sorts. But instead, the energy is given by these electrons falling down energy and releasing energy. So it's from a high energy state from complex 1 to a low energy state. And what these electrons do is it sort of release that energy, allowing for these active transfer against the concentration gradient. So there's actually more H pluses here than here. So in order for it to move against its will, so like imagine if there's one neighborhood where there's a bunch of suburban houses with a pool and a backyard, and on the other side it's cramped slums, right? Imagine if people from these suburbs continue to move into those slums, right? You're just going to get overpopulated, and it's the same way. But because of the energy released by these electrons moving down the chain, they're allowed to go against that, right? But this drives chemoosmosis, which is if you pack all of it on one side in the slum, right? There's a lot of items on one side, and then it wants to move back down naturally, right? Because it wants to move to an area with more space. And the same thing applies here, right? The protons are now in the intermembrane space between the membranes, and it wants to move back to a space with more, to an area with more space, which is the matrix. And these protons move through this structure called ATP synthase. And essentially what this does, it phosphorylates ADP into ATP by providing with energy from the electrons, as combined with actually the oxygen and the electrons, it essentially becomes water. So that's another thing you see with your urine and etc. That's one of the reasons why you have urine sweat, is because water is also released. Essentially, the energy helps, again, fuels this gradient, and then it allows for H plus to naturally move passively down without any energy input to sort of power server rotor, which again is energy again. So you're essentially you're putting a little input in to energy, and it's allowing for the creation of a lot of ATPs. These H pluses move by themselves down, and it creates ATP. It helps to phosphorylate ADP with a free phosphate group and becomes ATP, which is again energy that can be used at any time to their disposal. and sort of these are various ways in which uh, various different related steps to cellular respiration. Obviously, I would include these in separate areas to make it more chronological, but these slides have been changed. And so glycolysis can be reversed through gluconucleogenesis, neogenesis. And this is where pyruvate is just essentially reversed to produce glucose. And this occurs sometimes when you want to break down glucose, but then the energy is not needed. So you just return back through the liver, actually. Fermentation is very important. And what this does is essentially when there's no oxygen, the Krebs cycle can't occur. As you've seen, the carbon's lost to make CO2. It's not possible without oxygen. So in this case, when the electron transfer chain and the Krebs cycle aren't working, you essentially, the glucose is converted to pyruvate, but since it can't do anything, it needs to, in order to make it more productive and generate energy, there's an alternate pathway called lactic acid fermentation. And what this does is this converts pyruvate into lactate, whilst sort of inputting the electrons that were previously, the electron carriers that are previously unused. And this allows the glycolysis to occur again, because in glycolysis, NAD plus, like the electron carriers are given electrons, and this can occur if all the electron carriers are occupied. So this sort of replenishes it, generates a bit of energy at the same time, but this is actually very inefficient, and lactic buildup causes muscle pain and cramps and all of that you see in athletics. And PFK inhibition, so if you recall, phosphofructokinase is a enzyme within glycolysis, and what happens is PFK sort of regulates whether glycolysis actually occurs. If your body has plenty of ATP that's at its disposal, there's really no point in expending more energy in the energy investing stage in order to produce ATP if it's not needed, right? So it's sort of regulated by the concentration of ATP. If there's a lot of ATP, then it doesn't sort of function, or it doesn't catalyze the process. Whereas if there's little ATP, then it does. So here's just a summary of the energy accounting. So as you can see, there's 2 ATP from glycolysis and 2 ATP from the citric acid cycle. So again, these are prep stages essentially for the big generated one, which is 34 ATP from the electron transfer chain. And that essentially concludes our presentation. Thank you so much for watching, and hope you watch some other videos, and hope you enjoyed this presentation. So yeah, bye.